technical issues. So let's start the webinar. Today we are going to learn about the nerve impulse and conduction. Okay, I can do some slides, but I thought it's better to do something manually so I could teach you very well. So the first thing is, you know that the information in our body is processed via the nervous system. So for example, if you put your hand on a heat, you take your hand back suddenly, like you didn't realize it's a danger, but you take it as a reflex. So so it is because that the sensory neurons in your hands or a skin take those stimuli, send it to the nervous system, which it processes the information from the sensory neurons and give the effector organs the news that to take back your hand, like the muscles in your hand. And another example, like if someone calls your name from behind, you're turning and seeing them who is calling. It's because like the sensory neurons in your ear take up the information, send the information to the nervous system, central nervous system. They process the and send back the output to your muscles in your neck. Saying that to in your, if you zoom in and see what's really happening in your nerves, so there are opposite charges between two poles. You know, if there are opposite charges in two poles, there must be an electrical potential, which means a voltage difference. There must be a voltage difference. So you know, if there's a voltage difference, the charge particles can move here and there. So when electron flows here and the current flows like this, you know, this is the basic physics behind this. So normally, typically, the neuron is like this. There is an unequal distribution of ions inside and outside of the neuron. Like for example, can you see this? So assume that this is an axon. Inside the neuron or inside the axon, you have negative charge. Outside, you have positive charge. So it is an unequal distributions of ions. So you know there are sodium, potassium, calcium. Likewise, you have so many ions in your body. They have distributed unequally. So if the distribution is unequal, there must be a voltage difference across the membrane. It is called the membrane potential. Okay, the basic idea of this paper is you need to know there's an unequal distributions of ion. They are not same ions. There's not a neutral thing here. It's not zero. It has some potential difference here. So it calls the membrane potential. This is the basic idea of this paper. I hope you understand. Do you have any doubts to clear in this thing? I hope no. So we'll move to the next thing. So the first thing is you need to know the resting membrane potential. So as the name implies, you know, resting membrane potential means you need to know the definition. It means the membrane potential of a neuron when it is in rest. What do you mean by in rest? What do you mean by in rest? In rest means when there is no stimuli, no any other stimuli in the neuron. So it's typically it's doing its own work. It's typically rest. And so you need to know the definition and you need to know the values as well. It's minus 60 millivolts to minus 80 millivolts. The definition is the membrane potential of a neuron when it is rest. Rest means when there is no stimuli or anything. So it is maintained by, so these are the three main things you need to remember and you need to write when they ask in the essay questions. The distributions of ion concentrations inside and outside of the neuron. Okay, what do you mean by the distributions of ion concentration inside and outside of the neuron? It means, so assume this is your nerve cell. So the inside you have more potassium ions, the outside you have less potassium ions. Inside you have less sodium ions, outside you have more sodium ions. Sometimes you have calcium ions outside as well, but inside you have more chloride ions and more anions. What do you mean by A minus? A minus are anions. What are anions? Anions can be amino acids or any other peptides or proteins which contains negative charge. Some guys are outside. So the distribution of ion concentration inside and outside of the neuron is the main thing for the resting membrane potential. You may ask, why always inside is negative? Normally inside is negative, okay? It's the theory we learned in the previous paper, inside is negative, okay? Why it is negative? It is negative because of these ions. So there are some anions, and these two guys are the main reason for that. Inside, this guy is high, this guy is low, this guy outside, this guy is low, and this guy is high. And apart from those two main guys, we have these two chloride and anions. Anions means the proteins which possess a negative charge. And the second reason is selective permeability of plasma membrane to potassium and sodium plus. This is also a main reason for this. Okay, selective permeability of plasma membrane. Okay, this is the plasma membrane. You know it is a double layer plasma membrane. The selective permeability, what do you mean by selective permeability? It means that it will allow some ions to go out and come in. Okay, for potassium and sodium. Okay, here you have sodium leaky channels. Sodium leaky channels. So as the name implies, you can see leaky channels means it leaks sodium according to its concentration gradient. Now you know here you have a lot of sodium inside and a little bit sodium outside. So the leaky channels will leak the sodiums outside the cell according to the concentration gradient. What do you mean by concentration gradient? Concentration gradient means it does not use any energy. It will flow from high to low. Here we have a high concentration of potassium. Here we have low concentration of potassium. So it passively goes from inside to outside the cell. And we have sodium leaky channels. So you may ask, okay, we have sodium leaky channels. It can also go from inside to outside. Yeah, it can also go from inside to outside. It can also go from inside to outside like this. Here we have sodium leaky channels. So outside we have a lot of sodium. Inside we have a little bit sodium. So it can also leak from outside to inside according to its concentration gradient without using any ATP energies. But the scene is more potassium channels are open than the sodium channels. See here we have a lot of potassium channel opens than the sodium channel. So a lot of potassium goes out and only a little bit sodium comes in. This leads to maintain the resting membrane potential as more negative inside and more positive outside. So this is the second reason. Third reason, we have a special guy called sodium potassium pump. Sometimes we call this as sodium potassium ATP ACE. What do you mean by ACE? You have learned this. ACE means it's an enzyme. Enzyme means it's a protein. It's a protein, it's a channel protein, it's a membrane protein. Here is this guy. This is a special guy. This guy uses the energy. The name pump implies this guy uses energy. It carries 3 sodium from the cell to outside and 2 potassium to inside by using energy. Okay, so these guys are helping us to maintain the resting membrane potential. You know, resting membrane potential is inside negative, outside positive. So I'm just doing a quick recap to this. Resting membrane potential is the membrane potential of a neuron when it is in rest. What do you mean by rest? When there is no signal or stimuli. It is normally minus 60 millivolts to minus 80 millivolts. We can take it as roughly minus 70 millivolts. Okay, it is maintained by, so you all know, okay, inside positive, outside negative. Why? How does this happen? This happen because of these three reasons: distributions of ion concentration inside and outside of cell. So there's no equal ions inside and outside. There are different. So we have different amounts of ions inside and outside, and some special ions extra, like chloride and negatively charged anions like proteins and peptides are inside. So that's why the inside of cell is negative. And another reason is selective permeability of plasma membrane to potassium and sodium plus. So there are leaky channels. So this is according to the concentration gradient. Potassium goes out, sodium comes in. Okay, but it's okay. The thing is, more sodium channels are open than the potassium channels. So you need to mention that more potassium channels are open. Sorry. And the third guy is sodium potassium pump. This is a special guy. It takes 3 sodiums from the cell and puts it out, and take 2 sodium potassiums in, put it to in, using the energy against the concentration gradient. You can see here we have less sodium, but this guy is taking away sodium. So you know, if it means that against the concentration gradient means this guy need to use the energy. These guys need energy. Okay, so these 3 things are important. You need to know these 3 things and how does it happen. And some words are important. You can refer your handbook. The third thing is action potential. So you know, at rest, it will be like this: inside negative and outside positive. So when a stimuli comes, what do you mean by a stimuli? Or heat, a sound, or smell something? When a stimuli comes in, what happens to your typical neuron? Typical neuron be like this: inside minus, outside positive charges, like this. So when a stimuli comes, when a stimuli approaches this neuron, it become reverse. What do you mean by reverse? So inside you have negatively charged ions. Now it become reverse means inside you have a positive region, only for this region. So this is what happens in the action potential. Okay, so in the resting state, your neuron is like this. That means inside is negative, outside is positive. So when a stimuli arrives, it becomes reverse. That means a small region becomes positive inside and negative outside. So how does this happen? How? That means, so you need this graph to explain that. In the y-axis, you have the membrane potential. It is measured as millivolts. And the x-axis, you have time. It is measured as milliseconds. So here we have the resting membrane potential. That means if there is no stimuli, it will be like this at 70 millivolts. So when a stimuli arrives, it becomes slightly positive. It's going to positive. So how does this happen? Because when a stimuli comes, there's a channel called voltage gated sodium channel. It opens at minus 55. It means threshold. It means the least voltage to open a gate. So when a gate opens, when a sodium gate opens, sodium flushes in. Sodium flushes in rapidly. Then the polarity increases. And while you reach the plus 30 millivolts, the sodium gate closes and the potassium channel opens. So while the we can stop the sodium influx and the sodium will outflow. Okay, this is called repolarization, and I'll tell you the hyperpolarization afterwards. Okay, the thing is, at the resting state, you have your neuron like this, and when a stimuli arise, it reverses and the polarity changes and the action potential starts. The action potential has three states: depolarization, repolarization, and hyperpolarization. Okay, what do you mean by polarity? Polarity means change in charges. We have different charges. And depolarization means it's going near to zero, from minus to near zero or near to the positive. And repolarization means positive to negative. Okay, I hope you understand these things. Polarity means change in charges. Okay, then depolarization means we are moving further to positive. That means near to zero or near to positive. Repolarization means we are moving from positive to negative, again to the polarity. We are moving back to a minus. Hyperpolarization means we are moving further negative, from negative to further negative. Okay. Depolarization means negative to zero or positive. Repolarization means you are coming back to minus. That means positive to negative. Hyperpolarization means from the negative, you are moving further negative. So we'll come back to this chart when there is an action potential. So the neuron lies like this at resting membrane potential at minus 70 millivolts. When a stimuli comes, it changes its polarity by opening the calcium channels. Okay, if you open the calcium channels, sodium channels, sodium rushes in and make the inside a little positive. It makes a little positive, 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 and when it reaches minus 55 millivolts, it it opens other sodium channels and it will rapidly increase. So the polarity will rapidly increase, and when it reaches the plus 30, the sodium channel closes, so the sodium influx can stop, and the 30 millivolts will open the potassium channels, and potassium will go out. So the potassium will go out and turn back the neuron to the normal state, which is the resting membrane potential. But what happens here? This guy does not snap shut. This guy needs some time to close its door. Like it will close the sodium channel very slowly. The potassium will cell leave the cell further to further leave the cell, so it become hyperpolarized, like from minus to further, and it will finally it will come to the resting membrane potential by the sodium potassium ATPase. I said you, like this guy will maintain the resting membrane potential from the hyperpolarization. Okay, I'm telling you once again. So the action potential is the reverse of the membrane polarity. So at the resting membrane potential, we have minus inside. During the action potential, we have plus inside. This is because of the opening of sodium gates during the depolarization. and we can say the cessation of sodium influx can happen. At the same time, potassium channel opens and the potassium will go out. While going out, it's become to the resting membrane potential, but it's going further down because the slowly closing sodium channels. If it's slowly closing, the potassium will leave further, and it becomes more negative as ATPase, which is the pump, will use the ATP energy and restore back the resting membrane potential. I hope you have clear idea about this. If you have any doubts, can you unmute yourself and ask me, because it's a little bit confusing for you guys, and you'll get an MCQ definitely in this area. Can someone unmute themselves and talk to me whether it's okay or not, whether it's clear or not?
Yes, ma'am, it's clear.
Okay. Okay, then there's a refractory period. Refractory period means you cannot stimulate a neuron when it's in the action potential. So during the action potential, you have three phases: depolarization, repolarization, and hyperpolarization. During this time, you cannot send another stimuli to do this all over again. So this cycle will start here and end. After some time only you can send another stimuli. That is called the refractory period. That means you can't send another stimuli immediately to further process these things. That is called the refractory period. So why do you need refractory period? To ensure the impulse is going on one direction. Like if this is your axon, so in this area you have a action potential. So it will jump through like this. You have a small current like this. It will jump through like this in the forward direction. It will not go to the backward. So that is maintained by this refractory period to ensure the impulse is going on one direction. And you need to separately identify two stimuli. So that's why we have a refractory period. Refractory period means you cannot send a stimuli to a neuron immediately after an action potential. So the action potential doesn't recognize that stimuli because it is already in the process of doing these things. After finishing this cycle only, it can receive another stimuli and do all these things all over again. So that's the refractory period. Then we'll move to the conduction. So the conduction means, so here we have the we studied about the resting membrane potential, action potential. Now the action, so we said that action potential is generating like this. We are the sodium influx, we are the potassium efflux and other things. Now, so it cannot be only on the same location. It needs to move. It needs to move forward. So that is called the conduction. It's going forward. A series of action potential that move along an axon. So this is an axon in the neuron. It moving forward. It cannot be staying on the same location. It needs to move. That is called the conduction of impulse. Certain location. Okay, we studied that. So action potential occurs only in this location. Like here, because of the sodium influx, we have a little positive. So it needs to move forward. It is called the depolarization. Now, this depolarization spreads. It moves forward to the other locations and spread. While it's moving forward, the previous location will come to normal. That means repolarization. This action potential spreads to the neighboring locations and the initial location repolarize. That means, so here we have the action potential of the depolarization phase. Here we have the normal thing. Okay, this guy needs to move forward. While this guy moves forward and forward, the previous location will comes to normal. That means the repolarization. So while going forward, it becomes depolarization, and the previous location becomes repolarization. So this thing happens repeatedly to pass the information through the axon. That is the nerve impulse or conduction. Okay, I'm saying it once again, like a series of action potential moves along the axon. Along the axon, it needs to move. How does it move? That means, so in a particular area, we have an action potential. That means influx of sodium, the depolarization is happening. So it can't stay on the same location all the time. It needs to move forward. While moving forward, the previous location will come to normal. That means the repolarization. So it will repeatedly happen backward and repeatedly happen throughout the axon. So likewise, we have the current flow and the conduction. That means we are passing the information like the current. I hope you guys get this thing. And finally, in order to completion of this lecture, I had this synapse. So you assume that you are passing information like this, and from one neuron to another neuron, or from one neuron to a effector organ like muscle or a gland, you're passing details like passing at the terminal end, the terminal bud, you have this thing. Okay, this is called the synapse. That means at the terminal end, while the depolarization happens, uh, the depolarization voltage will open some calcium gates. Calcium will comes in and fuse with a vesicle, which is which has some neurotransmitters, and the calcium will help bind to the vesicles and bind to the presynaptic membrane. This is called the presynaptic membrane. This is called the postsynaptic membrane. The gap between this is called the synaptic cleft. Okay, so when this end is depolarizing, the voltage will open some calcium channels, and calcium will comes in and fuse with these vesicles, and this vesicle will fuse to the presynaptic membrane and release the neurotransmitters in it. So, and the neurotransmitter will come and bind to a receptor in the postsynaptic membrane, and these thing will open the sodium and potassium channels here. The sodium will comes in, potassium will goes out, like it will the depolarization happens here as well. Likewise, it's transmit the information. Okay, this is not in the webinar note. I just add I add this to the completion of the note. So the depolarization happens, calcium comes in, fuse with the vesicle, then the vesicle fuse with the presynaptic membrane, release the chemicals. The chemical will bind to a receptor on the postsynaptic membrane. That will open some gates, and depolarization occurs here. The cycle will happen same as that. Okay, then I hope you guys understand this, and I had some past paper questions. Can you all answer these? Okay, events that occur Events that occur after the membrane potential of a neuron is changed. Okay, you know, the membrane potential of a neuron is changed means what they mean by change. I told you the reverse means they are mentioning the action potential above the threshold value are given below. Sodium channels open and sodium outflow. Potassium channel opens and potassium inflow. Membrane becomes repolarized. Membrane becomes depolarized. Okay, I hope you all remember those things. In the action potential, we have something like this. This phase is called depolarization. This is repolarization, and the thing is hyperpolarization. Depolarization means something positively coming in. That means sodium in. Repolarization means sodium stops, potassium out. Potassium is going out. So they are asking the correct sequence of the above events. So the first thing is the sodium channels open and the sodium inflow. This is the first one. So the B is the first one. So you can cut off other answers which are starting from other letters. A. Here we have C, D. So these are not our options. So we can focus on these two things. Membrane become depolarized. Depolarized means something going towards zero or positive. So D happens. And then the repolarization means that the potassium channel opens and potassium go out. The repolarization. So the answer is the third one. Do you have any doubts in this question? This is a past paper question. Do you have any confusions in this thing? I hope no. So we'll move to the next question. Which of the following contributes for the maintenance of resting membrane potential of a neuron? We studied three things for the maintenance of the resting membrane potential of a neuron. So unequal distributions of sodium, potassium, calcium, and large anions inside and outside of the neuron. Oh yes, this is the first one we discussed. Unequal distributions of ions inside and outside. And the active transport of sodium out of the neuron and potassium into the neuron in 3 is to, that means active transportation, which use energy. Sodium goes out, potassium comes in 3:2 ratio. Opening of more potassium channels than sodium channels in the neuron membrane. This also we discussed. More leaky channels are open than the sodium. And the third one, the D option is transport of more sodium into the intracellular fluid of a neuron than potassium. We didn't discuss something like this, so this is not an option. And transport of more chloride ion from the neuron to the extracellular fluid. We didn't discuss anything regarding the chloride, so these are the wrong options. So these three are the correct one. I hope you guys understand this thing. The third question is select the correct statement regarding action potential of a human neuron. Regarding the action potential. Okay, so I'll do a recap on the action potential. This is the action potential. Depolarization, repolarization, hyperpolarization. Depolarization means sodium in. Repolarization means sodium stops coming in and potassium going out. Okay. Now we'll move to the question. Potassium influx into the neuron occurs during the repolarization phase of the action potential. So this is the repolarization phase. potassium going out. Not, it's not coming in. So this is a wrong statement. 5 milliseconds. No, in your handbook, because this is a question from 2018 paper, which is all syllabus. So this is not in your book, but better knowing those things. So this is also wrong one. The third one is sodium efflux from the neuron occurs depolarization phase of the action potential. So this is the depolarization phase during flushes in. So this is also a wrong statement. That means it efflux. They said that efflux. The fourth is it is a transient reversal of polarity of the nerve cell membrane. I told you guys this that during the resting membrane potential, the inside is minus, the outside is positive. So while a stimuli comes, it changes, it reverses, like the inside is positive, the outside is negative. So this is a correct one. The fifth one is immediately after one action potential, another action potential can be produced. No, it cannot be produced because of the refractory period. So you guys get a MCQ definitely in this area, and it is a bit more confused because of you guys are confusing between what's going in, what's going out. So that's why I chose this topic today. And you have an essay questions also. In 2018, they asked, explain how a nerve impulse is generated. You have two questions in this thing, how it is generated and how it is conducted. And you need to write how it's generated means you need to write the resting membrane potential and the action potential. So how does this resting membrane potential, how will be the neuron membrane? So inside negative, outside positive, how does it maintain? By those three things you need to write all these things. And the action potential, you need to mention that it is a reversible of polarity. It has three phases. During this phase sodium comes in, during this phase potassium goes out, during this phase this happens, like that. You need to explain these things in the generated, how it's generated. And while the conduction, you need to say that a particular area has the depolarization, it moves forward while the previous location comes back to normal. The repolarization happened, and the repolarization process are repeatedly happen. Likewise, it conducts through the axon. You need to write all these things, and it is an 80 marks because it's a part of a question. It is a B part of a question. Okay, I hope you guys understand these things. I'll